Good afternoon, everyone. Welcome to our Muscle Health Research Center seminar this afternoon. It's a pleasure to welcome Dr. Lauren Sparks to our seminar today. Lauren is from the Translational Research Institute in Orlando, Florida. I'll give you a little bit of background on Lauren. She tells me that she's a South Louisiana girl. She did her bachelor's degree at LSU, Louisiana State University, and followed that up with a PhD at Pennington Research Institute, which is in Baton Rouge, Louisiana. She then did her postdoc with Patrick Schrouwen at University of Maastricht in the Netherlands. And she's been an investigator now for 10 years at the Translational Research Institute. She comes from a family background in which alligator conservation is very big. And although her talk today is on human muscle, I can see she's very excited also about conserving animal species in Louisiana. Maybe we'll hear something about that. But anyway, we're excited to hear her talk entitled Metabolic Imprinting of Human Skeletal Muscle Cells with Exercise Training. So Lauren, if you wouldn't mind, please turn on your camera and unmute yourself, and we'll share your screen. And I'm excited to hear about this topic.
Thank you, David. I'm glad at least the alligator part made an impression on you. And I promise to tell everyone a little bit about that at the end, because David's right. I do get a grin from ear to ear just at the thought of an alligator. So I will share more of that later, but that is technically not why you have chosen to attend this seminar. So of course I'm blind to the audience, but thank you for taking the time to come and listen to me for about the next 45 minutes. This is also a very big passion of mine. The title is slightly different, but it's still on the same topic. It's just really, I wanted to emphasize that this is a focus on measurements around mitochondrial capacity in particular. I know it said metabolic imprinting, and a little bit of work I'm going to share. We're in the early stages of looking into some epigenomics in relation to how that impacts the mitochondrial capacity in these human muscle cells and tissue in the context of both diabetes and exercise training. So before I move past this slide, I know this is a room full of trainees or mostly trainees, so I will shamelessly put up my email address and just let you know that these are real pictures of sunny Orlando, pretty much year round. And this is a picture of our institute here. It's a standalone institute, and we're always looking for great talent, folks that are interested in metabolic research in the context of aging, metabolism, obesity, diabetes. So feel free to email me at any time if you're interested in maybe pursuing some work here. So I just like to make the assumption that everyone doesn't know exactly what I do. So I kind of split my research focus into two parts. I have two loves, in addition to alligators, two loves of both skeletal muscle and white adipose tissue. And I won't talk anything about my adipose tissue interests today, but I'll just tell you that I am doing very much a deep dive on single cell and some spatial transcriptomics in white adipose tissue in the context of exercise and aging. But on the muscle side, I also do a little bit of single cell work, but I'm focusing much more on this regulation of response variation in response to exercise in skeletal muscle in particular. And at the center of this, everything is focused on substrate metabolism, mitochondrial capacity. And because of the research area that I'm in and my background being in molecular biology, I do all of my work in human clinical trials, but everything is biopsy derived. So the skeletal muscle and the adipose tissue, my work is then performed in those tissues and then in the cells that are derived from that. So whether it be pre-adipocytes or the myoblasts. And I don't do this alone, obviously. In fact, I feel like I don't do much work these days other than write. And these are just a snapshot of the talented folks that I've had the great fortune of working with over the years, and some of which are currently with me. I also host visiting trainees. Mentorship is a cornerstone of what I do. I've highlighted the folks in red boxes that have contributed to the work I'll share with you today. So I'll just start by introducing folks to this, what we call the skeletal muscle niche. And you might be well aware of this, but it is integral to what I'm going to speak about today. So within that skeletal muscle niche, we actually have different types of stem and progenitor cells. We have the satellite cells, the classic PAX7 positive, and also CD56 positive. So highlighting those here. But within that niche, there are other cell types, progenitor cells, that I won't get into today, but some of which I do actually study alongside the satellite cells and the myoblasts. And these are PICs. I don't study these, but I do study FAPs, these fibroadipogenic progenitor cells down here in blue, as well as pericytes. And these can just give rise to non-myogenic cells. And that's for a discussion another day. But today we're going to talk about myoblasts, and these are the progeny of the satellite cells. And their main function is just to form to fuse with other myoblasts so that they can form these myofibers, or they can fuse with an existing muscle fiber and just donate their nucleus to that fiber. And then, of course, they'll just, in a healthy muscle tissue, they're going to return to a quiescent state and back into the progenitor pool itself and get ready to self-renew and again form with together with other myoblasts and myofibers under conditions of stress or injury. So the way we use these myoblasts, the way we first isolate them and use them, because people are using them in many different contexts. Regenerative medicine, that's not, of course, the context I'm looking at. Mine is much more with a metabolic lens. So the way we get at these is through muscle biopsies of the vastus lateralis. I don't really work with cells from other muscle groups, but of course you can, like rectus abdominis. So once these are isolated, they're quiescent, then get activated, and then we will expand and proliferate these in culture. And these are what we call myoblasts. We can then freeze these down and use them. I'm working with cells that I've had in our freezers for over 20 years. We can also, for experiments and assays, we can then take those myoblasts and expand and then differentiate those into myotubes. Now, one step that we do that not everyone does, but every bit of data I'm going to show you today, we do this immunopurification for CD56. Remember, I told you those satellite cells are marked with CD56 in addition to PAX7. So what that allows us to do, because this pool, if you haven't worked with these cells, it can be quite dirty. And as you passage them, you can also get takeover from fibroblasts. So what this sorting does is we find that it purifies that population so that we know we're working with just myoblasts. And then these, of course, would go on in vivo. They would form, well, in culture, they're going to form myotubes, and then they can become, of course, myofibers in vivo. And we also do, we don't do this every time, but we've gone through, painstakingly using gene expression to just confirm that these are, in fact, with rise and fall of particular markers like myogenin and myosin heavy chain that will rise. You get myo-D will decrease or decline throughout differentiation, to make sure that we're working with differentiated tubes. I hope you can hear this sound. This is 2D in culture, but this is actually a myotube that is twitching spontaneously. So these aren't 3D organoids, but they do have that ability to spontaneously contract. And I just thought Star Wars music was appropriate for that. So I do hope I didn't question David beforehand if the sound works. So hopefully you got to hear that. But if not, the take home was that these myotubes, even though they're adherent to plastic, they can spontaneously contract. And that'll come up a little bit later in my talk when we get into EPS models or electrical pulse stimulation. So again, I told you my focus and the real reason I work with these muscle cells is because back in the 90s, Bob Henry and Amira Clip very nicely showed that these cells are able to retain the metabolic phenotype of the donor. So this speaks to some epigenetic programming. But what they showed here and went on to show in a series of many experiments is that in these particular studies, these were cells taken from healthy non-diabetics and then individuals with type 2 diabetes. And they were here looking at insulin stimulation of glucose transport. You can see that and these are myotubes. So you can see in the white bars, healthy controls, they get that normal response that you would see in vivo during a glucose clamp, insulin clamp. But in those cells derived from folks that had type 2 diabetes, they had that blunted insulin stimulation of glucose transport. And they went on to then associate this with in vivo glucose disposal rates during a clamp, and just showed that the folks that were insulin resistant in vivo, their muscle cells were also insulin resistant. And this was the first time that this had ever been shown. So opening the door for what I think is a very valuable model in metabolic research. So sort of standing on the shoulders of Amira Klipp and Bob Henry, I did my PhD with Steve Smith, and we had some really nice studies showing that it wasn't just insulin stimulated glucose disposal. There were other aspects of metabolism that were also retained in these muscle cells. So what's being shown here is what we term an in vitro metabolic flexibility. So this just means that by using 14C labeled carbon, the substrate here is palmitate. So what you're seeing is a consumption of palmitate, so palmitate oxidation, that's on the y-axis, and this is called suppressibility because what we did is we titrated in increasing amounts of or concentrations of glucose, that's along the x-axis, and you get a suppression of fat oxidation. And sort of in the reverse, this is still palmitate oxidation on the y-axis, but here we're titrating increasing amounts, and these are low concentrations. These are not inducing insulin resistance. Titrating those in, you get this adaptability so that the cells are able to meet the challenge and oxidize. You give them more palmitate and they oxidize more palmitate or more fat. And then we associated this with the in vivo metabolic flexibility that we measured during the clamp. So we have change in metabolic flexibility here, as well as the change in insulin sensitivity, and basically just showing that in a group, I think this was about 60 healthy subjects, that the suppressibility or the flexibility in their muscle cells aligned or correlated with their in vivo metabolic flexibility. And later, I did some work whenever I was in Maastricht with Patrick Schouwen, showing that in individuals that were obese, that had obesity and or type 2 diabetes, that when we did some similar measurements, not oxidation, but giving the cells palmitate and measuring its incorporation into triacylglycerol, which is a good form of storage, an inert form of storage of that fatty acid, that in both the cells, the tissue and the cells, in the individuals with obesity and no diabetes, they had a higher formation or a better sequestration of the fatty acid compared to the individuals with diabetes. And then we related this with their glucose profiles in vivo. So just showing that those that had a better ability to store the fatty acid also had that had higher amounts of storage of fatty acid, also had higher levels of glucose. Now, one caveat to working with these cells, and why I pointed out earlier that we do the CD56 immunopurification, is that with increasing passages, they have a limited, these are not immortalized cells, so they have a very limited proliferative and myogenic capacity. And we do see differences by phenotype as well. So the more active and or lean, healthy folks tend to be able to retain their phenotype longer. And then those that are metabolically compromised, that phenotype tends to wane with passaging. I will point out that these are not, this is not our work. This is work from Sudeep Bajpai, who I worked with in the past, but these aren't CD56 immunopurified. But looking here, Sudeep did a great job of, this is a tremendous amount of work, but just looking at some snapshots of markers for type 1 fibers. I think this was a myosin heavy chain or staining that they did. Also looking at intramyocellular lipid and mitochondrial content. What you see here is that you have these differences across the phenotypes, with the highly active having the highest mitochondrial content, decreased in lean, and actually a little bit higher in diabetics. And what I want you to just pay attention to is that you start to see this relationship or this phenotype sort of wane with passaging. Not for everything. It looks like the active, like I said, can kind of hold on to their type 1 fibers in culture, whereas you pretty much lose it after passage 4 in these other groups. So it's just something to be aware of, and also why we limit the passage number and do the 56 purification. So I'm going to move into putting this in context of questions that we like to think about. I didn't show you all this data, so just take my word for it that in the literature, some of our own work, and I know work of Mary Ellen Harper and others, that mitochondria in the skeletal muscle of individuals with diabetes versus lean, They have an aberrant morphology, aberrant cristae formation. They're smaller in size. They have reduced oxidative and glycolytic capacity. And some studies show that there's reduced content. That's a huge debate in the field that I won't tackle today. But some remaining gaps that we are currently addressing and that I will show you today is that we wanted to know if there are impairments in muscle mitochondrial capacity and insulin sensitivity. But when we account for other confounding factors like BMI and cardiorespiratory fitness, we also really wanted to know if mitochondrial capacity is retained in culture. I showed you some metabolic aspects of these cells that are retained in culture, but up until now, mitochondrial capacity hadn't been very deeply assessed in human myotubes. And so is there a role for epigenomic programming? And if there is, can you improve some of these aspects with exercise training that we know are improved in vivo? Can you improve that and have it retained in culture in the muscle cells? So study design is very simple, straightforward. We had 3 different groups of highly active, obese, and individuals with type 2 diabetes. These 2 groups were completely matched for age, BMI, cardiorespiratory fitness. I'll show you that in a second. They underwent 10 weeks of endurance training and then repeated these assessments. That was just in the folks with, excuse me, with type 2 diabetes. Whole body level, we did clamps, we did VO2 max test, DEXA scan for body comp, and then specifically targeting the muscle in vivo, we did a measure of phosphocreatine recovery rate, which is muscle mitochondrial capacity. And then in the biopsies, we did a whole host of measures, some of which I'll show you, but not all, in the tissue. So first, I'm going to focus on the cross-sectional design. So I'll get back to the training intervention in a minute, but first I'm just going to show you cross-sectionally. Like I said, we had matched for pretty much everything between these 2 groups, which is our focus. The only differences you see is from the hashtag, and that's all focused on their insulin sensitivity and HbA1c, it being worse in those with type 2 diabetes. So to show you some of this data in figure form and not in table form, body comp, the take home I want you to keep an eye on with our highly actives in the sort of cream colored bars, those are the ones that are going to be the real comparator and stand out. We do not observe differences in terms of body mass and even body composition by fat mass, fat-free mass between those 2 groups that have obesity with and without type 2 diabetes. The only difference, as I said, in the table of the clinical characteristics, the only differences we see are during the clamp with insulin sensitivity, with it being lower in those with type 2 diabetes. I do want to point out real quick, this is a preprint that's online. If you want to dig into, like I said, I'm giving you a snapshot of some of the data, but if you want to see more of that data, it is available on bioRxiv. So really quickly, if you're not familiar with this measurement of phosphocreatine recovery rate, it is using a 31 phosphorus coil. I won't get into the big details, but we are measuring basically a suppression or a decline of PCR, and then its synthesis rate. So the way we achieve that is the person is at rest in the magnet. We have them kick against a resistance band. This is very, quick, within a minute. The phosphocreatine declines, we have them stop, and then we're measuring, this is the rate of synthesis. So the faster the rate, the better their mitochondrial capacity. It's a very highly reproducible measurement, and that becomes important later. So what we see here is aerobic capacity. VO2 max is, of course, higher in the highly actives, but not different between these 2 groups. And then, as expected, in vivo mitochondrial capacity also higher in the highly active, and not different between these 2 groups. This has been shown by Patrick Schrauen and others, but essentially these 2 measures are pretty much in lockstep. That's not always the case, but they do typically track together. Now, getting into the skeletal muscle focus, the more invasive measures, this is the team here that had a ton of fun doing so many measurements using the Ouroboros oxygraph. I know some folks use the Seahorse. We're using the oxygraph. It's just oxygen cathodes that are literally measuring oxygen consumption. This is an example of a trace, as we call them, under different substrates. So I'll show you the data in a second, but just to give you a bit of a debrief on what you're going to be looking at, this L1 is what we call leak, and that's just with either a pyruvate substrate or a fatty acid substrate, and then some malate and glutamate. Once we add in ADP, we're getting contribution toward oxidative phosphorylation, but just from complex 1. So we call that P1. P1 and 2 is where we've added succinate. So we now get complex 1 and 2 oxidative capacity. And then we will titrate in FCCP here at the end so that we're getting basically full electron flow across the membrane. And so that's what we call maximal capacity. And then we did go back and add in rotenone to take out complex 1 so that we're just looking at the flow through complex 2. But to simplify the data and just keeping it in context of, a very metabolic context and not a bioenergetics context, the take home here is that, again, our highly active group, no matter what the state is, in a carbohydrate supported, so this has pyruvate, here we have palmitoyl carnitine, but the rest, these measures are the same. These are in the permeabilized fiber bundles. So this is the tissue. We get, much like we saw in vivo, you're getting the active group being highest and then no difference between these two groups. Same story over here. So basically, regardless of substrate, there's no difference in the respiratory or mitochondrial capacity between those two groups. But when we look at content, we did want to address this because it is a debate in the literature, and it also depends on what measurement you're using. Here, we're using mitochondrial DNA copy number and citrate synthase activity and showing essentially the same thing, that those highly actives just have more mitochondria compared to these two groups. And again, no difference here. And when you normalize the respiration, it's not that the highly actives necessarily have higher or more higher capacity in general. It's probably because they just have more mitochondria, but those are obviously healthy mitochondria. So what we're really here to talk about are the cells. And again, there's more data in that paper that you can access online. So we did want to look at whether or not this was retained in vitro in their muscle cells. And so these are all the same subjects. And what we see here is pretty much what we saw in the fiber bundles. And so this is already normalized to mitochondrial DNA content, because in this context, you do have to have some sort of normalizer for these cells. And I'll show you the content differences on the next slide. But what we see is that when normalized to content, we are actually seeing no difference in these two groups in terms of respiration and having it highest in the active. It's significant over here with the pyruvate. It's showing some trends toward that here, but of course, not significant. But one thing that we did find interesting is that unlike in the fiber bundles, what we see in the cells is that the diabetics actually have higher mitochondrial content measured by both of these. It's not significant. We just had to do it separately with the assays. It's just the way it works. So technically not significant. But regardless, it does appear that the diabetics have more mitochondria. It doesn't necessarily mean, and this has been debated and it's been shown, I showed you that earlier from other studies, that diabetics, at least their cells, have more mitochondria, but it doesn't necessarily mean that those are healthy and intact and functional mitochondria. So to summarize the cross-sectional findings, sorry, my light went out. I didn't show you some of this data, but there are essentially what we conclude is there are no discernible differences between obese individuals with and without type 2 diabetes that are matched for other confounding factors like BMI, age, and aerobic capacity. And this is important because this actually has not been shown in the literature. There's a lot of data around this, but no one has actually reported on these, compared these two groups accounting for all of these factors. There are usually differences in aerobic capacity between those groups and or BMI. So what we see are no differences in content, capacity. I didn't show you this, but we have a lot of RNA-seq data to also show that there aren't really any differences between those two groups. We're highlighting that the lean active, of course, have enhanced mitochondrial capacity. This is also reflected at an epigenomic level, and they have higher mitochondrial content. I won't get into this, but we do see this intermediate transcriptional profile of mitochondrial capacity relative to the active and the obese. Basically, the individuals with type 2 diabetes appear to have a transcriptional profile that looks a little bit more like the highly active than the obese, which is interesting and something that we're following up now. So collectively, I think what we conclude in the manuscript is that this underscores this dissociation of mitochondrial capacity and skeletal muscle insulin resistance. And it's highlighting what we think is that you can have sort of this time course or a potential time course where insulin resistance can actually precede a deterioration of skeletal muscle mitochondrial capacity. But when we are looking at the myocellular mitochondrial capacity, the diabetics actually have, they do retain this reduced mitochondrial capacity despite having that higher mitochondrial content. So, and I apologize, my light keeps turning off. So late last year, we were invited by ESSR to do a review on in vivo versus in vitro training, if you will. So electrical pulse stimulation versus an in vivo exercise intervention. So I'm just going to pull a couple of snapshots from our work there. The revisions are pending, so hopefully that'll be published soon. But what I'm showing you here is just what I said, that you can actually look at the impact of exercise in two ways on these cells. You can look at it in the context of an in vivo training intervention and then isolate those cells pre-post and look at that. Or you can just perform the exercise intervention or the electrical pulse stimulation or other mimetics, such as pharmaceuticals, in vitro. What has not been addressed, so of course on this side, there are a few studies that have looked at mitochondrial quality and quantity in this context. Over here in the EPS model, there are lots of studies that have looked at quantity, not so much in terms of looking at quality. One thing I do want to point out, and a caveat, if anyone is currently using these EPS models, we have used them. I have tons of collaborators that use them. But one thing that I think gets overlooked a bit, and it's just something to keep in mind, is that there are studies that have been done that use EMG to assess an in vivo workload on the muscle. So cycling, you get an amplitude of about 0.14 millivolts and a frequency of about 80 hertz. A resistance training load in the vastus lateralis, this was also vastus lateralis, amplitude is about, call it 0.7 millivolts. If you move down into the in vitro EPS models, those workloads, you can already see the amplitude, it's not even millivolts. The amplitude ranges from 5 up to 40 volts. And the frequencies aren't so far off. They're about 100 hertz. But also those durations can be as little as an hour, 1 hour pulses, but some of them are up to 24 and 48 hours of continuous stimulation. So it's just something to keep in mind, because a lot of times people try to compare the in vivo to the in vitro and call that a true mimetic in vitro. And I think we just need to be careful with that. I won't get into this. This is just one of the tables that we have in the review, but just to show you that there are only 4 publications, one of which is ours, looking at the impact of in vivo exercise training on muscle mitochondrial capacity, or quality as we're calling it. And there are actually, we found 22 publications specifically focused on the outcomes of mitochondrial capacity and quantity using these in vitro or electrical pulse models. So, and also some of the pharmaceutical mimetics. So moving us over into exactly what I'm talking about, we want to know what happens with training in the muscle tissue and also the myotubes. So we went back to our study that I introduced earlier, and now we're going to focus on what happens in these individuals with type 2 diabetes post-training. So one thing, we have published this work prior, and it might seem a little tangential, but I promise it will make sense. So we published this back in 2018 now, but one thing we did is using this PCR recovery, this mitochondrial capacity measurement in vivo, because I care deeply about understanding response variation to exercise training. We used this outcome because it is so, the CVs are 4.5%. So it's very good in terms of knowing that you're getting outside of that technical error range. We used this outcome to split our groups into what we called responders and non-responders. So that's demonstrated here. So of those 17 individuals with type 2 diabetes, did 10 weeks of training. We had the folks over here that were, again, out, this red line is our technical error of the measurement. So these were classified as responders over here on the right, improving. And then we have 6 folks over here that did not improve and actually worsened. And this, there are other folks that have also shown this and seen this. So it is real. People actually do somehow decrease their mitochondrial capacity in response to training. I have some ideas around that, but that might be for another discussion. Again, this is all published, but just to show you, one big take home was that those folks that didn't improve or were the non-responders for mitochondrial capacity were also non-responders in terms of insulin sensitivity and glycemic control, in which they worsened their glycemic control. But both groups, importantly, improve their VO2 max. So the take home there is that it depends on your outcome, right? So it doesn't mean if you don't improve one endpoint that you're not improving something else. But what we really found interesting is that if we go into, this is just pre-training, this is not their post-training skeletal muscle tissue and cells. We took biopsies pre and post, but in the pre-training biopsies, we did RNA-seq and DNA methylation shown here, and the heat maps are basically, they're identical. And what you see is that even at baseline, before they've even done the intervention, they are clearly separating themselves. And we took the top end, we had 6 non-responders, and we just took the top 6 responders. So each line here, each column is an individual. And so, yeah, so they're distinguishing themselves already at baseline. And then whenever we did just a simple Venn diagram around the pathway analyses between the tissues and the cells, a lot of that overlap had to do with glutathione regulation, insulin signaling, and mitochondrial metabolism. And this is just represented here, digging into some of these pathways, specifically the DNA methylation here, RNA-seq here. It's superimposable between tissues and cells, and you get this nice reciprocal relationship between where you have higher DNA methylation, you had lower gene expression. We also did an integration of this data. So this is, these are the same data I just showed you in green, the type 2 diabetics. But we also did this in the highly active to just compare between the two, to try and have these two sort of the extremes in terms of the models. And what you'll see here is the way we did this, and this was with Marty Walsh, my longtime collaborator at Mount Sinai, who's the real epigenomics expert. He basically, what he did is he took the differentially methylated regions, that's this DMR over here, and then cross that over to the differentially expressed gene. So if it was a differentially methylated region, he only picked up the corresponding differentially expressed genes and integrated those two. And the main take home is that you do see these differences that are the same, the patterns are the same between the tissue and the cells. And whenever we did pathway analyses, a lot of that was enriched for mitochondrial capacity and muscle development. And this is just to show you, this is complete proof of concept. If we selected out a couple of genes, what you really want to see here is that the purple and green, which are tissue and cells of the active folks, and then gray and yellow, tissue and cells of the diabetics, they're segregating by phenotype, so that those tissues and cells really are retaining the integration of the CpG methylation and the RNA-seq signatures by gene. So this was enough for us to then take a look at some of the functional capacity of the cells. And I did show you cross-sectionally. So you're seeing that again here, I showed you tissue and muscle cell mitochondrial capacity cross-sectionally. Now it's taking just those diabetics and looking at them post-training. And so what I want you to notice here, and again, this has been published, if you take individuals with type 2 diabetes, they undergo an exercise training intervention, their muscle tissue mitochondrial capacity improves. And just looking at this maximal capacity, if we break this out into a waterfall plot, looking at the individual responses, it's really only one person that didn't improve or decrease their mitochondrial capacity, whereas everyone else improved in their tissue. So we wanted to see if that held up in the cells. You can already see that overall, there's no significant improvement like we saw in the tissue. So these are the muscle cells measured, same protocol, same way, pre-post intervention. But if we look at the waterfall plot, we do actually have some individuals, their cells are being imprinted in that way. So they are improving their mitochondrial capacity, we showed earlier in vivo, they're improving it in their tissue, and their cells are clearly being imprinted and also retaining that improvement at the level of their muscle cells. We do have others, however, that are not improving. and that actually is retained in their muscle cells. So we then moved away from individuals with type 2 diabetes. We thought, okay, well, maybe the presence of a metabolic disease is part of this non-response, or at least contributing to the variation of the response. So moving into another study that I'll show you in a second, we've published on many times, but this is a group of healthy, lean, and overweight individuals, mainly comprised of women. We did same measures of body comp, VO2 max, and muscle mitochondrial capacity in vivo, in tissue, and in cells. We've previously published this work, and over here on the left-hand side, you'll see the in vivo measures. On the right-hand side is the tissue up here. So we show this nice, we had pre-post training, and we did again have an active group as a comparator. We get a nice increase in vivo as well as in the tissue. And then this was just last year, my postdoc Rochelle went back to this study and looked at just the cells of these individuals. So not the tissue. Down here, we're looking at the cells. I apologize, it's quite blurry. But on this left-hand side, this is the same data here, just looking at it within those individuals that were then measuring their cell respiration. So they all improve. There are no non-responders, essentially. There's not much variation, just maybe the magnitude, but they all improve. And we see the exact same thing over here in these individuals in their cells, they're all improving. So that variation, or that the folks that didn't improve in the diabetics, we don't see that here. So this again is a very small sample size, but just to recap the training responses, we do see an overall improvement in VO2 max in individuals with type 2 diabetes, but there is this variation in vivo mitochondrial capacity and insulin sensitivity. Pre-training epigenomic profiles in both the tissue and the cells highlight key differences between responders and non-responders. This is something we're currently following up. We also see response variation in vitro in these same cells in these individuals with type 2 diabetes. But whenever we look over to a study, again, small sample size, but whether it's because they're healthy or it was a much more intensive training intervention, we don't see this variation. All of the cells seem to improve in vivo, in the tissue, and then that's maintained in their cells. So the take-home message there is that, skeletal muscle mitochondrial content versus capacity remains debatable in type 2 diabetes. This is part of our paper that's on bioRxiv, but you do need multiple assessments of capacity and content. In some of these populations with diabetes, the insulin resistance actually, it seems to precede the development of blunted mitochondrial capacity. Whenever we want to start looking at this impact of exercise training in vitro in muscle cells, we can turn to models of electrical pulse stimulation. We just have to approach those with caution because they aren't going to fully recapitulate what we're doing in terms of training and the actual load on the muscle in vivo. And of course, something that's near and dear to my heart, and I think it's starting to get more widely accepted, is looking into the variation in this exercise response, because it does exist, certainly in terms of muscle mitochondrial capacity, not just in vivo, but we're seeing this retention of a response variation or even a blunted response in the cells. But the caveat there is we need much larger cohorts and more in-depth studies that can combine both the functional and the epigenomic assessments in the same cells and in the same individuals. So this is something that I'm currently undertaking now. I was hoping that I would be able to share some of those data with you. Unfortunately, the powers that be, this is what happens when you're part of a consortium. We're embargoed and we can't share any data prior to completion of the parent study. But I will tell you, I'll just spend the last few slides telling you about the project. This is a central focus of my research, and I'm doing this together with Joe Humard at ECU, and I told you my collaborator Marty Walsh at Mount Sinai. This is an ancillary R01 to a large NIH common funded study called MotorPack. It's looking at the molecular transducers of physical activity. So essentially these are healthy adults. It's almost 2,000 adults that are being studied, and these are broken out into groups. These are my numbers for our study, so this is not the 2,000, but we have all of these groups are represented in the larger study. So we have a group of highly trained resistance athletes, highly trained endurance athletes. We're getting about 40 folks in each one of those. They're not undergoing an intervention. They're just doing the study. We are collecting their muscle tissue and then establishing myoblasts. We also have, after they do an acute exercise bout, so we're just getting the biopsy pre-exercise bout, then they undergo in the sedentary group. We have folks that are randomized to resistance training or endurance training, and this is 4 days a week that they're doing. We're getting 300 individuals, so it's 150. It's a massive amount of work. It's 5 years, and we're almost done. 300, so 150 per group. And then we do have 75 individuals that are not, they're in the control group. So the goal here is to, one, you can see by the numbers, create a repository of these cells. And that's for individuals like you. So this is open to the public. Whoever wants to have access to these cells, you write a proposal and you submit it, and you can do your own studies. Something that we're doing, that's the primary focus, is to take these cells, and these are the endpoints that we're looking at. I just showed you mitochondrial capacity. That's what we're doing at Advent Health. At ECU, they're doing insulin-stimulated glycogen synthesis and insulin signaling. And at Mount Sinai, they're doing DNA methylation RNA-seq. And the important thing to know about the epigenomics, that's actually being done in the tissue, the muscle tissue of these same individuals as part of the larger MotorPack study. This is a consortium, so it's a massive effort. We're down here in Florida, but we have 6 other sites that are helping us collect these cells. That's how we're able to get such large numbers. So we have Louisiana, Texas, Colorado, California, and North Carolina. And this is where we are to date. We also have another talented postdoc, John Noon, who is helping with this work, but we're extremely proud and happy to report that we have finished collections on all of the highly active individuals. We even exceeded those collections. So we have about 80 subjects split evenly between those two groups. We're conducting work in those now. We're about halfway through on the sedentary individuals. So we have 2 more years to go to collect those. And we are using some of those for the current experiments just in the pre-training, so no post-intervention assessments now. And what we're looking at specifically is not just a basal comparison of these assays for insulin action and mitochondrial capacity. We are actually doing this at, in a basal context and also in the context of a metabolic stressor, something that Joe and I have talked about because he's worked with these muscle cells for years as well, is that you're not always able to see these phenotypic differences in a basal state, but it's really whenever, and I think that's the same in vivo as well. It's not until you really put a stress on the muscle or on the cell that you start to see the ability to either adapt or not. And what I mean by adaptation is what I showed you, that old work that I did with Steve Smith, where if you challenge the cells with a substrate, say palmitate, can they rise to that and adapt and increase their oxidation, or do they have a blunted response to that? And so what we're doing is to induce insulin resistance in these cells. And again, we're comparing sedentary to athletes, right? So the hypothesis is that the athletes will be able to adapt to even this high amount of palmitate and not have blunted insulin action. And then we are doing a mix, a more gentle mix of palmitate and oleate for 24 hours, again, to test that adaptability, both with fat oxidation and a carbohydrate supported oxidation. And we're looking at DNA methylation RNA-seq. This is just the most data I can show, which is we're doing 15 per group here, and we have, selected out the subjects from this larger cohort and matched them for age and BMI. But what we are getting are the differences, the expected differences in VO2 peak and knee torque in terms of strength versus endurance between those two groups. So I will just close by, of course, thanking all of the folks. I showed you their pictures earlier, but some of the folks I didn't show you their pictures: Brett Goodpaster, Steve Smith, Rich Prattley, who have been just fantastic mentors throughout my time and continue to support and give me advice along the way, and all of the amazing trainees that have contributed to this work and still contribute to this work. My collaborators, Marty Walsh at Mount Sinai, and Mary Ellen Harper at University of Ottawa, and Joe Humart at ECU, and of course the funding. And as promised, I will finish with some alligators. So this is actually my now 8-year-old, but my daughter who was 3 back in 2018, she's holding an alligator hatchling here in her hand. She is gloved, so we're using proper technique. But I was telling David earlier that another way that I link alligator conservation to some of what I study in the world of epigenomics is that the alligator and many other reptiles are perfect examples of epigenetics in terms of their sex determination being temperature dependent. So you change the temperature and you change the sex of that species. And this is my daughter 5 years later. This was just a few weeks ago. She got to come full circle, and this is the hatchling. And then years later, you do get to release some of them back into the wild as part of the conservation effort. And we do take that tape off of their mouth before they're released into the wild so that they can survive. And yeah, that's all I have for today. I'm hoping we can have some good discussion and questions. Hope I did all right on time.
Great job. Great job. Thank you very much, Lauren. That's great. I love that. Not just the alligator, the data too. Very, very, very interesting data. Okay, folks. So as per usual, if you could just type your questions into the Q&A, please, and we'll get to those momentarily. Maybe I can start off with a question because I'm fascinated by the responders and non-responders, and you said, you know, if you want to, we could discuss that later. So that's what I'm asking you about, these individuals who responded and not responded, despite the fact that they in all groups increased their VO2 max. The change in oxidative capacity and the change in insulin resistance didn't match in some of those, and you believed you had a theory of some kind. You want to share that? Or
yeah, so I mean, we do spend a lot of time thinking about this, and you know, every context and every cohort is different. I'm resubmitting a grant, expanding this study now, because one thing we were missing was a control group there to be able to track the change across time. So there is data out there from Jane Rush to suggest that diabetes itself is part of the non-response. That these ambient hyperglycemia, these high levels of glycemia can impact the response to exercise in terms of improving glycemic control. That makes sense. John Kerwin has also published that. I think Jane also still thinks, and she and I are good friends and colleagues, and we continue to discuss this, but she does have data showing, I think, quite nicely that in individuals with type 2 diabetes, she uses the same measurement of mitochondrial capacity in vivo, the PCR recovery rate. And what she has shown there is in the individuals with diabetes, it actually comes down to the ones that don't improve also have a blunted oxygen diffusion to the muscles. So that could certainly play a role in their ability to reap the benefits and the improvements. And as you know, diabetes comes in all different flavors, right? So one diabetic is not like the other one, and they have different issues. And so that makes it a little bit messy. You also have medication use. We did have folks, we washed them off of their medication for that study. So they were hyperglycemic. And so we can't ignore the fact that that played a role in their blunted response. So long story short, I think the best is to go back to a sort of healthy non-diabetic population and establish the response. But what I'm most interested in, and this is where I was sort of saying, oh, we can talk about this later. I put it into my grant. We'll see how it does. There's literature on the rate of the response, right? Because whenever we do all of this, it's a snapshot. It's a pre and it's a post. And wherever you end up at that magical post measurement, it could be a 6-week intervention, it could be a 10-week, 12-week, that's how you're classified. But there is data on VO2 max to show that I think it was measured weekly for 6 weeks, people are all over the map. You have people that within a week, they go up, and then they plateau, and then they might go up again. Or you had some that even declined at the end. You had some that declined right away and then went up. So I think this rate of the response is going to be critical. And this is what I've built into it, that it all depends on where you're catching these people. I mean, I'm sure anecdotally, all of us can relate to that, right? I mean, some of us, and I know Mary Ellen has great data on this from the diet weight loss perspective, that you have fast responders and slow responders. I mean, it could just be a matter of time for some people. So I think that's an important aspect of response variation that has been overlooked. So it's something that I'm working into, and it does depend on the outcome. You have to be really good at measuring what you care about.
Yeah, no, good points. So I'll get to the questions in the chat in a moment. I just have one kind of controversial question to ask, and that is, you said that insulin resistance precedes the mitochondrial capacity changes. And I just,
Potentially.
Potentially, okay. Because of course, some people believe that the mitochondrial, fragmentation and abnormalities and stuff like that lead to lipotoxicity and then insulin resistance and stuff like that. So I just wondered if you wanted to weigh in on that just a little bit, without.
Yeah. I mean, if I'm honest, I mean, you don't write this in the paper. It's just based on that data. I mean, my gosh, my postdoc and I, and Mary Ellen Harper is part of this paper, we had to do a real deep dive in the literature. And if anyone has done this, dug into the literature on insulin resistance and mitochondrial capacity in diabetes, it's all over the place. You can strongly support either point of view. So where we landed with our data up against the rest of the data was what I put in one of my take-home messages, was that the one thing we found, and this is credit to Katie for finding this, is that people weren't always controlling for VO2 max, especially. There tended to be differences between those two groups when you looked at individuals with obesity, with and without, whether it was insulin resistance or diabetes. There tended to be differences either in VO2 max or some age differences, but it was mainly VO2 max. And we know that VO2 max tracks with mitochondrial capacity. So there was no way to really say for sure, well, what if they didn't? What if they had accounted for that? Would they have still seen the difference? So that's kind of the best I can say. I mean, I believe the data on both sides. I've seen some really strong data to say both ways. And so I think what it comes down to is, I think I even said it like that, in certain type 2 diabetes populations, because I just think they're all different. And if you don't account for every single variable, then it's hard to make conclusions. So I do feel confident in our conclusions, where when we do account for those things, clearly there's no issue, or there's no difference. I shouldn't say issue. It's blunted compared to highly active, but it's equally blunted in both. Whereas the individual's diabetes clearly are insulin resistant and have blunted glucose disposal rates compared to those without.
Mhm.
Doesn't mean those that don't have diabetes won't ultimately get there. And that, again, this is depends on when you measure them, right? Week later, maybe it could have been a different story.
Right. Fair enough. Good enough.
That's my political answer.
Okay, sure. Anthony Shime asks, your excellent data suggests that there are satellite cell adaptive changes with obesity, exercise, etc. Is there a way to perform in situ fixation on human muscle biopsies as done in mice, which would retain satellite cell quiescence to assess their adaptive changes by RNA-seq?
Yeah, so I think Anthony is referring to probably the same paper. I'm thinking of a paper that he might be referring to, and this was probably came out 5 or 6 years ago, where they did show very nicely that DNA methylation profiles, they compared mouse versus human. When they did this fixation in situ, the transcriptional profiles changed immediately. As soon as you pluck those cells out, it changes. And I should mention that actually as a caveat that I didn't. And so that's why we actually, in the beginning, I called them satellite cells, because in the niche, they are satellite cells. But we know as soon as we remove them, I actually call them progenitor cells, because we know their transcriptional profiles change. DNA methylation profiles are rock solid. Those are steady. But I guess a short answer to your question, Anthony, I think it's a great point. We have not done that. But I do think it's an important point that should be raised, that those profiles do change. For us, we're always comparing it, like I showed you the responders, non-responders, we're at least comparing within or, across phenotypes, and they're all collected the same way. So while the transcriptional profile is not necessarily reflective of that satellite cell in vivo, whatever has changed would have changed equally across the groups.
Mhm. Yeah. Okay, Mary Ellen Harper says, great seminar, Lauren, thanks. Can you comment on the possible mechanisms explaining the differences in transcriptional profiles in the type 2 diabetic that were intermediate between the obese and the trained groups?
This is because Mary Ellen's on the paper and she knows the data. I didn't actually show you this data. I just put it as a summary bullet. This was fascinating and shocking to me. And so that point may not have come across as well just because I listed it as a bullet. What she's referring to and what we saw is that, we have these PCA plots of DNA methylation and RNA-seq in the tissue across these three groups, the highly active, the individuals with obesity with and without diabetes. You've got exactly what you would expect, what you saw with the mitochondrial capacity. You've got highly active over here by themselves. And then there's not as much separation between the two obese groups. You move into the cells, and for whatever reason, the individuals with type 2 diabetes, the PCA plots, their profiles are actually shifted over closer toward the highly active. And the obese are off by themselves. One big thing that I didn't highlight in the clinical characteristics table is that highly active group, it's all males. The obese group is all females. That's a huge issue there. So we have sex to wrestle with. And then the diabetics are at equal mix. It's not what I expected. So now we're seeing that the cells from individuals with diabetes have higher mitochondrial content. They have transcriptional and DNA methylation profiles, at least when we look at them with mitocarda and pathway analyses, anything related to mitochondrial metabolism, it's much more like the highly actives and as opposed to the individuals with obesity. And when we do all these comparisons, things are blunted at a transcriptional level in the obese cells compared to the active and the diabetics. And it is shocking and surprising. I'm not really sure I understand it. It's just the interpretation is that individuals with diabetes are not necessarily hardwired in that way to have deleterious mitochondrial capacity. It's probably something that's happening more in vivo with maybe these hyperglycemic levels that the muscle tissue is constantly bathed in, as opposed to there's something about the individuals with obesity that seem to already at the cellular level be blunted or almost pre-programmed to not have blunted mitochondrial metabolism capacity. Yeah.
Mhm.
So
Very
sorry, I couldn't show that data. There just wasn't enough time, but it is some of it, not all of what I just shared, is in the preprint.
Yeah. But it is the type 2 diabetic population that shows the greater number of non-responders, right? There's
Yes.
in the healthy individuals, there were, I've forgotten the measure it was that you showed, but there were no non-responders in something, or at least only one out of several, most of them. And the type 2 diabetics had many more non-responders, right?
Correct.
Yeah.
And I think there's just more heterogeneity there in the diabetics than you see in these healthy.
Okay, so one more, I think, just a technical question I had. You made a point of sorting by CD56 positive cells, and that seemed to be very important. I just wondered from a single biopsy, I mean, how's the yield? What is it? Is it substantial that I guess you're going to plate them and grow them anyway, right? Is that what's going to happen there?
Yeah. So we had to get this really dialed in for that for the large study we have going on now, because we had to train 5 other sites how to do this, and we had to get everything standardized. So you can, from a single biopsy of about 50 to 75 milligrams of tissue, we are freezing down 22 vials, 10 at passage 2, 12 at passage 3, 500,000 cells per vial. Those are not purified. So that's just straight, we get it, we digest, we expand, we freeze. It's not until, and this is important for folks that want to access those. So those are sitting there because for our experiments, we're only using 1 vial at each site. So there are still more than 15 vials per subject that you can access. But what we do is when we thaw that vial, we expand and then we do the sort for the purification for the CD56. And then we move on from there. We do always freeze back some sorted ones just so that we don't have to keep doing it. But I would say, sort of this is anecdotally, you are saving yourself at least 1, if not 2 passages by doing the purification.
Right. And you have the population you want. You have the myoblasts.
Just the myoblasts.
Right.
And it does make a huge difference in terms of the differentiation. And
Yeah. No,
Yeah.
sounds very good. Sounds very good. Okay, I'm not seeing any more questions popping up in the chat. So I guess what we should do is call it a day on a Friday afternoon.
yeah,
I want to thank you very much.
this is fun.
Stay with me for a second. Folks, thank you for joining us today. And we'll see you at the next seminar. Seminar's over, and I'll see you next time. Wait with me.